that Ron invited to this meeting have been talking to one another about the science and medicine of ME/CFS. Now we turn to you, the patients, the caregivers, the relatives and friends. And in this community symposium, we want to convey what we've learned about the molecular basis of ME/CFS. In doing so, we're telling all of you that we think you are among the most courageous people on the planet. Every one of us feels the urgency to cure this horrific disease. With that heartfelt introduction, let me move on to the Itaconate Shunt. I'm fortunate to be joined in this work by a bunch of smart scientists and physicians. I love these people. You should too. Okay, now a short history of Itaconate. This is from a review by the O'Neill Lab in Ireland, and introduces the idea of the molecule called itaconate. I'm only going to focus on a couple of the points in this nice timeline. One is that IRG, or ACOD1, has two names. They're historically important because IRG1 stands for immune responsive gene 1. And I was attracted to this gene because it is immune responsive. And we have the feeling, as Joshua emphasized in his presentation, that many cases of ME begin with an infection. So these other points that I want to emphasize in the timeline are that IRG1, or ACOD1, has been shown to be protective in both viral infections and bacterial infections. And those are all results from only a few years back now, 2018. And then I put on the slide something that was published after the timeline, which is the first paper I read on the itaconate pathway. This is also from the O'Neill Lab, but it's also from the Artyomov Lab in St. Louis in the US. These two labs are competing with one another about itaconate. And I've been in science a lot longer than those guys. And I want to suggest that they should be best friends, not competitors, because they taught me about itaconate, and I think the world needs them both working together. So then only in 2021, at this meeting actually, was the first presentation I made on the Itaconate Shunt hypothesis for ME/CFS. I gave that talk 6 months after Chris Armstrong, Paolo Macalini, Ron Davis, and I had been working on this idea. All right, so my plan now is to introduce the Itaconate Shunt hypothesis because it's evolved over the years as we've collected more data and as other scientists have designed their own tests of this idea. So let's look at this step-by-step process. Now, there's nothing on this slide now. What I want to do is step-by-step reproduce the whole hypothesis so you see it growing from the basics to the final or the current view. So if you've ever taken a biochemistry or a biology course, you've studied the Krebs cycle. You've studied things that happen inside the mitochondria. 95% of everything that's going to be on this slide when we're done happens in your mitochondria. The Krebs cycle is one of those things, sometimes called the citric acid cycle. And the basic idea of the Krebs cycle is that it is a machine for converting food into reducing equivalents, that is, molecules like NADH. So there are 9 steps in the Krebs cycle, and many of them, the ones that are indicated by the blue arrows, produce what we call reducing equivalents. Those reducing equivalents are really electron pairs that have been extracted from the food molecules. There's also that orange arrow in the Krebs cycle, which is a stage when you can make one molecule of ATP directly. But the most of the ATP that's formed is formed using the energy that's now incorporated in the NADH, the reducing equivalents. So the next stage is the electron transport chain. NADH transfers those electrons to electron transport chain complexes in the inner mitochondrial membrane. And that energy is used to pump protons across the inner mitochondrial membrane from inside to outside. In turn, that energy is used to convert ADP into ATP by another complex in the electron transport chain, which makes ATP based on the energy that you created by pumping protons. So there's a big membrane potential across this membrane, and there is a huge chemical potential as well. That energy is used to drive the synthesis of ATP. And you all know, I'm sure, that this ATP is used by cells to do their work. This is the work that God and evolution decided these cells have to do. And all that work is powered by conversion of ATP to ADP. Even if you didn't take biochemistry, or if you did, you probably don't know this next mitochondrial function. And that is that in addition to all this important energy producing work, the Krebs cycle is the source of succinyl-CoA for the synthesis of heme. Everybody knows about heme because it carries, it is the basis on which hemoglobin is formed. And of course, hemoglobin carries oxygen in red cells around the vascular system to all the tissues of the body. But heme is important for another reason, which is why I'm emphasizing it here today. It's an essential component of several of the complexes in the electron transport chain. So it's not surprising, really, that the heme synthesis pathway takes place largely in the mitochondria, because it has to supply heme to mitochondrial complexes. Now we're ready for the Itaconate Shunt. The Itaconate Shunt starts at cis-aconate. Cis-aconate is converted to itaconate, then to itaconyl-CoA, citramylyl-CoA, and finally, it is reconverted to pyruvate and acetyl-CoA, which, as you probably know from that biochemistry course, are able to re-enter the TCA or Krebs cycle. But it's really the failure of this Itaconate shunt to make energy that makes it important for ME/CFS. You can see immediately that if I divert a lot of the carbon that's usually going around the Krebs cycle out into this alternative pathway, that I will not go by the blue arrows or the orange arrow in the Krebs cycle. In other words, the Itaconate shunt is diverting carbon away from making energy. In fact, the Itaconate shunt makes at most one NADH. And so you can see that it is highly inefficient to go via the itaconate shunt. But it's even worse than that. It's worse because itaconate and itaconate-CoA are toxins. They also have important anti-inflammatory functions, which is what's dominating the literature right now. But it's important to realize that they have negative consequences as well. And so that's what I'm going to talk about largely today. The itaconate shunt produces these two toxins, and the production of them and the concentrations of them depend on two enzymes. ACOD1, or we call it CAD for cis-aconitate decarboxylase, and clybal, a molecule that is responsible for converting citramyl-CoA back into pyruvate and acetyl-CoA. So in some sense, I think an important sense, your disease baseline is, on this theory, determined by the ratio of clybal to ACOD1. That's because the more ACOD1 you have, the more of these two molecules, itaconate and itaconate-CoA, you're making. And on the other hand, the more clybal you have, the faster you remove itaconate-CoA from the system and put it back into useful biochemical work. So the higher this ratio, the better for your disease baseline. And I mentioned that the cell work is, for example, doing muscle contraction or thinking. That's what's going on down here in the lower right. ATP is required to drive all of that work. And so, for example, if ATP falls, it's harder to do that kind of work. And of course, if the electron transport chain is not getting what we call reducing equivalents in the form of NADH, it's not doing the work that is required to make ATP. So you'll have less ATP. Fortunately, ADP speeds the work of the Krebs cycle. And so that happens. I wanted to tell you that itaconate is a toxin because it inhibits SDH, and itaconate-CoA is a toxin because it inhibits ALAS. That's here. So those red bars with dots at the end imply inhibition of those two processes. So importantly, as I say, ADP speeds the Krebs cycle, and it does so by activating the enzyme isocitrate dehydrogenase. And what you see here is a potential explanation for the phenomenon of PEM. And so I'm just going to go through that, and then we'll move on to the data. So if you activate when you're either thinking or exercising, the work of converting ATP to ADP, you now have a signal in the form of ADP that speeds the Krebs cycle. It speeds it by activating this enzyme. You're going to make more NADH, and you're going to be able to keep up with the ATP consumption by having more ATP production. The consequence of that is that if the itaconate shunt is in place, is that more carbon from the Krebs cycle is going to go out into the itaconate shunt. That means there'll be more itaconate and more itaconate-CoA. And so if there's more itaconate-CoA and you inhibit the production of heme by inhibiting ALAS, their consequences are that downstream electron transport chain will not work as well. And interesting to me is that this happens within a couple hours after you inhibit ALAS. But the existing electron transport complexes already in the inner mitochondrial membrane will participate in electron transport and producing ATP for days, even up to a week. So I think that it's possible that this can explain the long sought reason for the delay in PEM. All right, that hypothesis, the diagram I just showed you, makes several predictions. One of them is that in ME, there'll be decreased oxygen consumption. In some sense, this is not a prediction, this is a post-diction, because we knew already from Kara Tomas's work and Julia Newton that there is reduced consumption of electrons of oxygen in ME. You can see that in their data published in the early 2020s. Even for moderate ME/CFS, there's a substantial decrease in ME/CFS. Excuse me for a sec. I have been talking for 3 days straight, and I usually don't do that. Okay. So decreased oxygen consumption is a prediction and also an experimentally verified one. Another prediction of the hypothesis is that because you're decreasing NADH production by shunting cis aconitate away from the Krebs cycle, you're going to produce much less NADH. And so the NADH to NAD ratio will decrease. And this was confirmed by our collaborators at the Bateman Horne Center who collected the samples, by Amanda Oy, who's at Stanford in Ron's lab, and by Eric Schmidt and James Cox, who are our colleagues at the University of Utah. And they ran the mass spec that did this analysis. And what you see here is that the mean represented by that open circle in the two is significantly lower in the ME/CFS patients. So this is a confirmed prediction of the Itaconate Shunt. And then another prediction is that the mitochondrial membrane potential should be decreased. And again, this is work of Amanda Oy at the Stanford Genome Technology Center. And she showed using a potentiometric dye that enters cells and enters the mitochondria, that the mitochondrial membrane potential is decreased compared to healthy controls in ME. That's done in the way that Amanda told all of us during the 3-day scientific symposium. So this too is a prediction of the Itaconate Shunt. You're going to decrease the mitochondrial membrane potential, and that is what we measure. This is a pretty complex diagram, but I just wanted to briefly show it in order to explain how the next experiment works. What we're going to do now is measure the mitochondrial redox efficiency, by which I mean how many electrons, how many electron pairs in the form of reducing equivalents are the mitochondria delivering to the electron transport chain. So you've probably heard of complex 1 in the electron transport chain. Here's complex 2, that they are working in parallel to deliver reducing equivalents in the form of NADH or FADH2 to the electron transport chain. And the common receiver of all those electrons is CoQ10 or quinone. And the quinone is converted to a quinol form, which delivers its electrons to another familiar name, cytochrome C. And cytochrome C has a particular redox potential that we can take advantage of because we have dyes, in this case a tetrazolium dye, that can accept some of the electrons from cytochrome C. And when it does, a purple precipitate is formed. And that's what we're going to measure in this next experiment. So here we're measuring the accumulation of reducing equivalents. That's what's plotted on the vertical axis, so that you can think of this as increasing NADH, delivering electrons to the electron transport chain. And the horizontal axis represents time during this experiment. Also, this experiment was designed and run by Amanda Oy in Ron's lab. And as you can see, really exciting for me anyway, is that the ME patients are producing many fewer reducing equivalents. It's really important to realize that this experiment is done with exactly the same fuel concentration. It's done in a plate where we've permeabilized the cells and we're supplying just one fuel at a time. And in this case, this experiment is done with the fuel being cis-aconitate. And you can see there's a significant decrease in the production of reducing equivalents from cis-aconitate in ME. So this too is a prediction of the Itaconate Shunt hypothesis, because we're diverting cis-aconitate away from the production of reducing equivalents. There are fewer in ME-CFS. So the last prediction on my list for today is that this decrease in ME-CFS production of reducing equivalents will be most profound at cis-aconitate. And you can see from this table, where I've listed some of the fuels that Amanda tested, that the largest decrease in reducing equivalent RE production in ME is at 32% or maybe even 33%, and is highly significant. Whereas for other fuels, it's still a decrease, but not as profound as it is in ME-CFS. Now, many of my colleagues, and probably some of you as well, will be thinking that the previous 4 predictions are predictions that will be made by many hypotheses for ME-CFS. But I think that all those hypotheses will have difficulty explaining this table. And the fact that cis-aconitate is the point in the Krebs cycle where the Itaconate Shunt takes off, and it's the point which is most profoundly affected by lost reducing equivalents, is to me the best evidence I have at the moment for the activity of the Itaconate Shunt in ME-CFS. The problem isn't further upstream. It's not in the delivery of glycolytic carbon from glucose to the Krebs cycle. We know that because we're using the same amount of fuel for both sets of experiments, the experiments on the healthy controls and the experiments on ME cells. This is done on PBMCs, peripheral blood mononuclear cells that were obtained from patients and healthy controls. Now, despite making these measurements, we do not yet have gene expression data or metabolomic data pointing directly to the Itaconate Shunt. We've tried to do that, and we haven't seen an increase in the ACOD1 gene expression, and we haven't detected cis-aconitate or, itaconate and itaconyl-CoA. Now, I'm an optimist, and so I think this is a detection problem, and it's caused, I think, by this disease being cell autonomous. What Ron and I always say about this is that if it's cell autonomous, and only a few cells are sick, it's going to be difficult to make this kind of measurement. So we were surprised and disappointed that there were not increases in ACOD1 expression in these ME PBMCs. But if you're a philosopher of science, you'll recognize that this is how theories evolve asymptotically toward the truth. And so we're now working on alterations in this hypothesis in which ME is characterized still by an Itaconate shunt, but the shunt is not controlled by the usual expected mechanism of transcriptional control, but is instead controlled by post-translational modification. Okay, so the Itaconate shunt, as I've shown you, is a theory of ME/CFS, which, if correct, has multiple disease-causing consequences. It short circuits the TCA or Krebs cycle by diverting carbon into an alternative pathway that doesn't make anywhere near as much NADH or FADH2. This decreases the pumping of protons in the electron transport chain and decreases the production of ATP. Low ATP in the mitochondria and in the cytosol of all cell types is, I think, what causes ME/CFS symptoms. And the more cells that you have running the Itaconate shunt, the worse will be the disease. And the more itaconate and itaconyl-CoA you produce, because you can't clear it with Clival, that will make those symptoms worse. So in some sense, the production of these two toxins is an important part of the mechanism of ME/CFS if the Itaconate shunt is the underlying pathology. And as I said earlier, when ADP speeds up the Krebs cycle, as it normally does when cells increase work, then the increase in flux around the TCA cycle will, because the percentage is the same, increase the flux into the Itaconate shunt and produce more itaconyl-CoA. I think that's the source of post-exertional malaise, or PEM. This also explains why PEM is a transient, that is a very slow transient, of course, but a transient which you can eventually recover from, because this speeding up of the TCA cycle and the slowing of heme synthesis are driven by the exercise itself. So when the exercise itself stops, or the thinking itself stops, the enzyme Clival can continue to remove the itaconyl-CoA that was built up during the exercise. But you then return to the original steady state, which was determined by the ratio of Clival to ACOD1 in the resting Krebs cycle. So let's see. We don't yet know how the control of ACOD1 and Clival are maintained, but that's what we're working on, and we think it's important. So I think we're on a valuable research path, and I thank you for your attention.
Thank you so much, Robin. That was a great presentation, as always. We really appreciate your work.